Alright guys, so we're going to officially start this unit with our first lesson. Now we did kind of start it before you went on break, and you had a little bit of an intro over microbiology. So really what we're going to do in this unit is we are going to cover a couple of organisms and organism-like viruses that would be kind of considered in this microbiology unit. We're not going to focus on all of them in this unit. So what we're going to focus on today is going to be prokaryotes. And some of it should be review from what we did back in the fall semester, and some of it's going to be information that maybe you haven't seen before. So just really quickly, what is microbiology? It's just a study of microorganisms. And so microorganisms could be anything that we really can't see with the naked eye. Sometimes we just simply refer to these as microbes. Now, most commonly, we're probably going to think of bacteria when we think of microorganisms, but the archaeans could fit in this group, as well as some of our other kingdoms. So protists, many of them are microscopic, and they would fit in this category. Fungi. Now, while some fungi are not microscopic, there are some like yeast cells that are. And then it could also include other infectious agents like viruses. And viruses are not living things, but because they can be disease-causing and they do have some characteristics of living things, they would kind of fall under this microbiology unit. Now, we're not going to go into all of the history. You saw some of that in the video. But way back when the microscope was first discovered in the 1600s, they just kind of used basic lenses to look at tiny things. And while Leeuwenhoek was not the first person to invent a microscope, he was the first person to see these microscopic organisms. And so he was just looking at pond water on a very basic microscope, and he could see these little tiny organisms, and he named them animalcules. I think there was another, I don't know if it was him or someone else that named them wee beasties. But early on in the 1600s, 1700s, this was when we were first able to start to see and study these microorganisms. Now, we've come a long way with our microscopes and electron microscopes and things like that. And so we introduced the unit with the video and an article, and there are some certain concepts that I want you to retain from that. So I want you to be familiar with the concepts of spontaneous generation and biogenesis. Remember that spontaneous generation was the idea that living things could arise from non-living matter, like microorganisms could arise from gravy, or the flies could arise from meat. And then it was through a series of experiments where this idea was disproven. So it was believed for a long time, and we know that science and our ideas can change through experimentation. And so through the works of Redi and Spallanzani and Pasteur and some others, the idea of spontaneous generation was disproven. So I do want you to be familiar with those particular people and how they kind of disprove spontaneous generation. And so what we believe today in our current accepted theory or idea is biogenesis, which is the idea that life comes from life. And we remember hopefully from the cell theory back first semester that one of the statements of the cell theory is that cells come from other living cells. And so obviously it took some experimentation to get there, and today this is what we widely accept. So as I mentioned earlier, microbiology includes any tiny organism, but in this particular unit, in this lesson specifically, we're going to focus on the prokaryotes, and then in our next lesson we're going to focus on some other non-living things, but it's still infectious agents known as viruses. So a little bit of background on prokaryotes. A lot of this should be review from first semester, but remember that prokaryotes are cells that do not have a nucleus, okay? So they lack that nucleus structure, that membrane-bound nucleus that houses the DNA, and they lack all membrane-bound organelles. If you don't remember what those are going to be things like mitochondria, chloroplasts, lysosomes, Golgi bodies. Now, it's important to understand that even though these cells do not have the organelles, it doesn't mean they are unable to perform the processes. There are bacteria that can perform cellular respiration, and there's bacteria that can perform photosynthesis. They just don't have the little compartments to perform them in. Now, what they do have, like all cells, they have a plasma membrane on the outside. They have ribosomes for protein synthesis. They have cytoplasm, which is just that filler, the liquid-like, jelly-like component. And of course, they have genetic material. Now, the difference is their DNA is not housed in a nucleus. It is just found in the cytoplasm in an area known as the nucleoid region. Most of your prokaryotes do have a cell wall, and as we get into the kingdoms, we're going to see that that cell wall is going to divide our prokaryotes into one of two groups. It's one of the defining characteristics of our two domains. But most of your bacteria do have cell walls, and most of your archaeans do have cell walls. Now, there could be some other structures that they have, like a capsule. A capsule is a sticky substance that's found on the outside of the cell wall. And not all bacteria are going to have capsules, but those that do, it can be used for protection. One of the things it can do is help them survive in a host because it helps them kind of avoid our immune systems. They could have these structures called fimbriae or pili, and these are little tiny structures that help them stick to other surfaces. Now, when you look at a picture of the fimbriae or the pili, they look similar to a structure known as cilia in a eukaryote, but they are not the same thing and they do not have the same function, okay? So these fimbriae or the pili, they are an attachment structure. It helps them attach or stick to something else. Now, they do have flagella sometimes, and flagella is going to be a tail-like structure for movement. Eukaryotes can have flagella as well, but eukaryotes will have a different structure to their flagella. The flagella in a prokaryotic cell are going to be a little bit simpler in their structure, but in both cases, in both types of cells, flagella are meant for motility or movement. Now, when it comes to your prokaryotes, they are unicellular. They live as single cells. That means one cell, but sometimes they'll form colonies, and if you ever try to grow bacteria, like on a petri dish, one little tiny bacteria will reproduce rapidly and give rise to that colony, which is that bunch. So when you see a colony growing on a petri dish, you're seeing possibly hundreds, thousands, or millions of bacteria in one little area. We can't see the individual bacteria with the naked eye, but we can see them as the colonies. Now, typically, there are going to be some exceptions to the rule here, but typically your prokaryotes come in one of three shapes. They could be cocci, bacilli, or spirilla. And these right here, I believe, are the plural form. You could also see it as coccus, a US, bacillus, a US, or spirillum, a UM. And so one of those is singular, one of those is plural. But cocci are round or little spheres, and the way I remember this is you see the O, C, C, those are round letters. And so that's kind of what your cocci bacteria look like. Now, this isn't written in the notes, but I just want you to make a note that sometimes they can be singular, or they could come in pairs, or they could come in clusters, or they could come in chains. So this is just describing the shape of an individual bacteria, but sometimes they'll kind of live together as a group. Even though they're individual organisms, they may kind of cluster in different ways. The same for the bacilli. Bacilli are rod-shaped. They always remind me of little peels or little capsules. And so you can see that they also can come in groups. And then your spirilla, they are typically either going to be a spiral or a helical shape. And then there's some other ones that aren't mentioned here. Once again, these are the three main shapes that you will find, but you can see some other variations depending on the bacteria. Now, the genome, this is a term that we've learned before, hopefully you remember, but genome just refers to the complete set of genes in that organism, or essentially the genetic material. And so when it comes to the prokaryotic genome, in comparison to a eukaryotic cell, they don't have nearly as many genes. Okay, so they have smaller genomes. They have about 1,000th the DNA of a eukaryote. Because they're simpler cells, they don't need quite as many genes to be a living, functioning organism. Now, one thing that varies between eukaryotes and prokaryotes is a prokaryotic cell only has one chromosome, And so if I asked you to draw a chromosome, you would probably draw something like this. And this is what a eukaryotic chromosome looks like. It's what we call linear. It has a top and a bottom. Remember, we have these chromatids that are held at the centromere. But a prokaryotic chromosome doesn't look like that. It is one big circle. Maybe if you remember when we did the prokaryotic notes back in the fall, in comparing them, they just have one chromosome instead of many, and their one chromosome is circular. And once again, it doesn't reside in a nucleus, it resides in an area known as the nucleoid region. Now, this particular chromosome has all of the information necessary for that bacteria to be a living, functioning organism. It carries all of the genes that it needs to survive and work as a living cell. Now, one thing that your prokaryotes have that eukaryotes don't typically are these little tiny circular pieces of DNA called plasmids. Now, plasmids are not necessary to survival. In fact, they don't have to have them, and they can lose them. But if they do have them, there are some genes on the plasmids, and so it could give them some additional traits, like the ability to conjugate or antibiotic resistance. And so plasmids are something that are used quite a bit in genetic engineering because they're dispensable and they can be exchanged from one bacteria to another. And so these plasmids are just handy little extra pieces of genetic material. Now, when it comes to reproduction, prokaryotes are asexual, which means you essentially have one parent cell that will divide into two. And the process by which they do that is known as binary fission. So it literally means kind of split into two, binary two, fission is split. And so the way they do it is they essentially just copy their DNA and divide in two, as you see over here. It's a little less complex than, say, mitosis. Mitosis would be part of a eukaryotic cell division process. And so binary fission is just literally an asexual method. So your daughter cells, based on reproduction, are going to be identical to the parent cell. And they can do this very rapidly. If you ever have grown bacteria on a petri dish, it doesn't take long to see those colonies forming because very quickly one becomes two, two becomes four, four becomes eight. And because it's asexual, the bacteria don't get a lot of variation, or they don't get any variation through reproduction. However, it doesn't mean that all bacteria are identical because there are some variation generating processes. So down here we have a few examples of how bacteria can generate some variation. The most common way is just by random mutation. We know that when DNA gets copied, mistakes can be made or chemicals could cause a mutation. And so you could have some random changes to the DNA. And then we also have these three processes here. Now, what these three processes are is they are examples of gene transfer, ways in which DNA could be transferred or inserted into a bacteria from another source. And so all of these can kind of shake up that genome a little bit. Now, transformation is just the uptake of foreign DNA. So they could be exposed to DNA in their surroundings. Maybe a bacteria around them died and was broken up. And so they can somehow kind of assimilate or take in this DNA through their cell membrane. In fact, we take advantage of this and we do transformation experiments to kind of make bacteria do this. But they will naturally take in foreign DNA from their surroundings. And when they do that, they could obtain the traits in those genes that they take in. We do transformation experiments like with glow genes, or we put human genes into bacteria. So we definitely take advantage of this natural process of bacteria. Another way is conjugation, and this is where the plasmids can be exchanged from one bacteria to another. If they have a certain gene, they're able to form this specialized pilus called a sex pilus, and through that pilus, they're able to pass plasmids from one bacteria to another. And then another way that they can do gene transfer is through transduction. So just like we have viruses that infect our cells, there are viruses that infect bacteria cells. And as they go from one bacteria to another, they could carry some genes in the process. So here's just a few images to show you what these lateral gene transfer processes look like. Once again, transformation is just the uptake of foreign DNA. So you can see here there were some plasmids in the surroundings and they're able to bring them in, or you could have pieces of DNA out here in the surroundings and that bacteria can take it in. For transduction, it requires a virus to transfer that genetic material, and conjugation, they're able to form a sex pilus. Now remember, none of these are reproduction. These are just some ways that bacteria can kind of shake up their genome and get some variety. All right, when it comes to metabolism, prokaryotes are very diverse. They can obtain nutrition and energy in different ways. Now, most are going to be heterotrophic, which means they're going to have to acquire both their energy and their food or their organic compounds from some other source. They could either be decomposers or they could be consumers. But we also have autotrophic prokaryotes, and autotrophs come in two forms depending on where they're getting the energy. So autotrophs could be chemoautotrophs, and we're going to find these in areas where you don't find light. Like they could be at the bottom of a body of water, they could be in a cave, they could be in a hydrothermal vent. But chemoautotrophs are able to get energy from inorganic compounds from the earth, and they're able to convert them into food. So they don't get their energy from the light like a photoautotroph would. Now normally when we think of autotrophs, we think of photosynthetic organisms, those like leafy plants that are able to turn light into food. And there's bacteria that are able to do this too. Now they don't have a chloroplast to do it in, but they are able to take energy from light and make food. So there's a wide variety amongst all of your prokaryotes. Same thing for energy production. Some of our prokaryotes are anaerobic. Now this is that process where they don't use oxygen. They can do glycolysis and fermentation, and they can make a small number of ATP at a time. And then there's other prokaryotes that are able to do aerobic respiration. Remember, it's way more efficient. With oxygen, we can generate a lot more energy. So they don't have the organelles like the mitochondria to do it in, but they are able to do these processes. And so depending on the bacteria or depending on the archaean, some of them are aerobic, some of them are anaerobic, and some have the ability to do both. When it comes to your classification of your prokaryotes, this should be a review for you, but we have two domains that include prokaryotes. We have domain bacteria and domain archaea, and then their kingdom names are the same. Now, I do want to give you a heads up that if you're ever looking at resources, at one point, these were called kingdom eubacteria with an EU. So some resources still have that old classification. And then these were called archaebacteria, and they've since kind of changed those names just to the same as the domain. So if you were ever to come across eubacteria or archaebacteria, just know that that's what we are today calling bacteria and archaea. And so we're going to take a look at the differences between these two. Your bacteria are what we commonly think of as bacteria, the ones that we kind of deal with in our daily lives. They're everywhere. And then we have archaeans that are extremophiles. They live in very extreme environments. And so based on some nucleic acid studies, when they compare like DNA sequences and certain RNA sequences, it's believed that archaeans are actually more closely related to eukarya, but we're going to talk about them here because they do fit under that classification of a prokaryote. So starting with domain bacteria, these are your most diverse and widespread. These are what we would consider common bacteria. They're very tiny, so they're measured in micrometers. This prefix micro here means 1/1,000,000th. Okay, so when we're talking about micrometers, they are 1/1,000,000th of a meter or smaller in length, up to about 10 micrometers. So if you were to take a meter stick and you were to divide that into 1,000,000 pieces, this is about how many of those pieces your bacteria would be. Now, one of the defining characteristics of domain bacteria is their cell walls contain the substance known as peptidoglycan. Now, peptido refers to proteins and glycan refers to sugar. So your peptidoglycan is just going to be a protein and carbohydrate matrix, and that's what's making up the cell walls. Now, one of the ways that we can identify organisms within domain bacteria is through a process called a gram stain. This is a very common process, and it helps us put these bacteria into one of two groups. Now, there are going to be some that don't fit into either, but most of your bacteria are either going to be described as gram positive or gram negative. Now, what this is it's a staining technique, and so depending upon the composition of their cell wall. Now, in-domain bacteria, they all have peptidoglycan, but some have more than others. And so gram-positive bacteria have a very thick layer of peptidoglycan, and when they go through the staining process, they stain purple or violet. And so I remember that gram-positive are purple with lots of pep. Okay, so positive purple, lots of pep. And we're going to take a look at some of these here in a minute, but bacteria like strep and staph both fit under this gram-positive category. And then gram-negative, they have a much thinner layer of peptidoglycan, so notice they do have it. They're bacteria, so they have peptidoglycan, but they have a much thinner layer, and they often have this outer layer beyond the cell wall called a lipopolysaccharide. And so this is kind of a fatty, sugary type of substance. And so they do have the peptidoglycan, but not nearly as much. And so when they go through the staining process, the purple actually washes out, and they end up staining pink or red. And so I like to go with the red because purple started with the P up here. So I just remember negative, less pep, and red. So we have positive purple, lots of pep, and negative red, less pep. And so the gram stain is an important process. Like if you go to the doctor and they take swabs because you're sick and they're trying to identify what you have, one of the things they're going to look at are the shape that we mentioned earlier, the cocci, bacilli, or spirilla, and they're also going to do a gram stain because immediately we can put bacteria into different groups based on their shape and how they stain. It's also important to know their gram stain because when it comes to antibiotics, bacteria will respond differently to the antibiotics depending on the type of cell wall that they have. So it's definitely an important piece of information that we need when we're looking at treating disease bacteria. Now, some of the major groups, if we were to look at, the phylogenetic grouping and phylogenetic trees, we can put these into kind of 5 major groups. We have proteobacteria. A lot of these are gram-negative, so they kind of stain red. Some that you may recognize, and you're going to be researching some of these in your activity here shortly. Proteobacteria. Your gram-positive bacteria, most people recognize Streptococcus and Staphylococcus as strep and staph. There's some others that fit in this category. So they have those really thick peptidoglycan walls, and they would stain purple. Another ecologically significant group are the cyanobacteria. Now, these used to be referred to as blue-green algae, but they're actually bacteria. But these are very important because they're photosynthetic. They're actually believed to be the ancestors of chloroplast. They also are a part of a mutualistic relationship with fungi, and they form lichens, so these are pretty ecologically significant. Now, chlamydia bacteria are actually a parasitic group. They are a type of sexually transmitted disease, so chlamydia is a bacterial STD. Spirochetes, they have that spiral shape, that spirilla or that helical type shape. They move by kind of using this corkscrew movement. And one example of them is Borrelia burgdorferi, which is actually a disease-causing spirochete that you're going to look at in your activity. So here's just some general examples of bacteria that we see in our lives. Some of them are good, some of them are bad. It just depends on the specific bacteria. All right, so our next group, domain archaea, once again, they are extremophiles, or they live in extreme environments, places where a lot of other living things would not survive. Really high temperatures, really salty areas. They could live in anaerobic conditions where no oxygen is present. And so they're going to live in places where they may be some of the only life there, because typically high temps or high salt or no oxygen would be deadly to a typical cell. And so they have these special characteristics that allow them to live in these areas. They've evolved certain, specialties. And one of those things are some special lipids. So They may have cell walls, but because of their diversity and environment, they may not have a specific structure in their cell wall. So we just simply say that they do not contain peptidoglycan. So that's a big characteristic in dividing the domains, is that bacteria have PEP and archaeans do not. Now what you will find are some unique lipids. They have these branch side chains in their cell membrane lipids, and they may have cell walls, but there would be different structures that they would be made up of. So once again, they're tiny. We see those measurements in micrometers. Some of them are a little bigger than your typical bacteria, but they're all going to be microscopic and tiny. Now the major groups here, we have 3 major groups, and that we could put them into other phylogenetic groupings, but our 3 main groups are halophiles, thermoacidophiles, and methanogens. And so halophiles live in really salty conditions like the Great Salt Lake. Thermoacidophiles can live in extreme temperature or pH. So you would find these like in hot springs, places that temperature would normally kill other living cells. They can live in very acidic environments, and they do have some special lipids in their membranes. And then we have methanogens, and so these are the ones that produce methane as a byproduct. These are found like in swampy areas. You know, methane stinks. Methane can be like when you pass gas. And methane, a lot of animal intestines contain these methanogens because there's not a lot of oxygen in there. And so what they do is they take carbon dioxide and hydrogen, and they're able to produce methane when they metabolize. And so like cows release methane because of these bacteria that they have in their intestines. So here's some other examples of places where you might find some methanogens. Once again, pretty extreme, not a lot of other life in these areas because of those conditions. A lot of times enzymes would denature, and so many other cells would not be able to survive. All right, we're going to kind of finish up with some important roles of prokaryotes. And honestly, bacteria specifically get a bad rap. A lot of times when we think about bacteria, we think, ooh, gross, bad, wash your hands, where's the hand sanitizer? And the truth is that bacteria can be bad, but the benefits of these microorganisms far outweigh the negatives. In fact, we would not be able to survive and live without all of the beneficial bacteria that we have in our ecosystems. So they are major components of food chains and food webs. Once again, remember that some are autotrophic and they're producing in areas. They could also be heterotrophs like decomposers, and they have major roles in nutrient cycles. Nitrogen fixation is something that really only a special group of bacteria can do when it comes to living things. We have to have usable nitrogen, and we can't rely just on the atmospheric methods. We need these bacteria to help put nitrogen into a usable form. They're also important in decomposition, and that's not just important in the nitrogen cycle, but also in the carbon cycle. And because we have photosynthesizers and respirators, they're going to be important roles in that carbon cycle as well. Many of your bacteria and microorganisms are part of symbiotic relationships. That could be mutualistic, commensalistic, or parasitic. And so just a few examples of mutualistic relationships. We have many mutualistic bacteria that live on us and within us. Our digestion is heavily dependent upon microorganisms. We have bacteria on our skin that help kind of maintain an acidic pH, and that kind of helps keep other bad microorganisms away. Those cyanobacteria form mutualistic relationships with fungi. Now there are some that are parasitic, and these kind of give bacteria a bad rap, and so they can, break down tissues or harm the host by kind of using them as a nutrient source. And so in that sense, there are some bad ones. And then, of course, there are pathogenic or disease-causing. Now, this isn't limited to just bacteria. So in this particular lesson, we're focusing on pathogenic bacteria or pathogenic prokaryotes. As of now, archaeans aren't really connected to disease too much, and it would be my guess in that we aren't really exposed to them. Typically, humans are not going to be in those extreme environments where many of your archaeans live. So they're not really the disease causers. It's mostly going to be that domain bacteria. And when they are disease causing, they cause disease in one of two ways. Either A, they are physically breaking down your tissues. So some bacteria could use your tissues or other organisms' tissues for food. And so in the process of breaking them down, obviously that's harmful to us. Or they could be disease causing because they're producing toxins. And those toxins in turn cause a disease. So some of them have endotoxins where we ingest the bacteria, and then when our digestive system breaks them down or lyses them, it releases these toxins, and these toxins can cause diseases like food poisoning. They can also produce an exotoxin, and these can be very, very deadly. They can be released by living bacteria. You've probably heard of tetanus. You may have had a tetanus shot, or things like the plague or cholera. These have caused many, many deaths in the past. Now, luckily, we have a lot of treatments for bacteria in today's world, but there were times when bacteria just caused millions and millions and millions of deaths. And they still do cause deaths in our society today and in areas that don't have a lot of updated medicine. Now, I do want to clarify, once again, they get a bad rap, but the majority of them do not cause disease. As I mentioned earlier, these are the ones that we think of, and we think of bacteria as bad, but in the grand scheme of things, most of your bacteria are not disease causers. And then some of our last roles, they have very important roles in industry. We use bacteria to create products and to make our daily lives easier. We use them to make certain foods like yogurts and cheese. We use them in the process of fermentation, like to make alcoholic beverages. We use them in biotechnology a lot. And here's the thing, I mean, there's so many bacteria and they reproduce so rapidly that no one really cares if we kill them in the process. And so bacteria are very easy to use in a laboratory setting. We use them to make recombinant DNA. We use them in these transformation experiments. We can move genes around with the help of those plasmids. And so we've got bacteria that pump out human proteins. We have bacteria in labs that are making human insulin and human clotting factor and human growth factor because we can insert human genes into bacteria and then get them to make those proteins for us. And that way we're not having to take them from another source. And they are literally the human protein because we've inserted the human genes. And so we use bacteria a lot to pump out products like medicines. We can use them to create vaccines. And then another way we use bacteria is through bioremediation. So we use bacteria in our sewage treatment processes. We've biogenetically engineered some bacteria to clean up oil spills and other waste type scenarios. So there's actually quite a few beneficial roles. And I added this, it's not in your notes, but I thought it was a great summary of prokaryotes as a whole. It kind of talks a little bit about their structure and their reproduction and their roles and their diversity. And so I would just encourage you to kind of take a look at this little graphic. It's got a lot of important information on one little picture. And that is going to wrap up our first lesson on microbiology and prokaryotes. We're going to follow up this lesson with some practice, and then we'll be taking a quiz here in a couple of days. And so hope you guys had a great winter break. As always, I want to remind you that I'm here if you need me or have any questions, and I hope to see you soon. Bye, guys.